Hello, my name is Cody, and I am a biological sciences major. And today I will be discussing the research I've been doing in the Gross lab, assessing functional changes in genomic organization in the hemoglobin complex of Astyanax cavefish. Our story begins in the Sierra de El Abra region. This is a limestone region in northeastern Mexico. There are over 30 caves in this region. However, we're going to be focusing specifically on Pachon Cave. Now, when we think of cave environments, we're thinking of very extreme environments with the lack of sunlight, low levels of nutrients, and, relating to our research, less dissolved oxygen than in surface watersheds. So what we see in this figure is that specifically in Pachon, there's a 64% reduction in the amount of dissolved oxygen compared to surface watersheds in the area. Now this is important when looking at the Astyanax morphotypes because we see a few key differences such as loss of pigmentation, regression of eyes, but we also see differences in organization of hemoglobin genetics between the fish. Now, why hemoglobin? Well, hemoglobin is the molecular transporter of oxygen in the blood. So when contemplating lower dissolved oxygen levels, this may relate to the hemoglobin genetic differences that we see. And when we compare the surface fish genotype to the cave fish genotype, we actually see that the surface fish does have more hemoglobin genes than the cave fish. So the surface fish has 19 hemoglobin genes compared to the surface fish only having 13 hemoglobin genes. Now, you might ask, wait, fewer genes? This may be counterintuitive because when thinking about evolution, one would assume that having more genes leads to having more proteins that can help out in a more extreme environment or just a different environment in general. But what we see, number one, is that caves are very stable environments. And so even though there's less dissolved oxygen, there's less variability in dissolved oxygen levels. So it's not fluctuating a lot. And this is very similar to another interesting and relevant story that was told by Balsarod et al. In 2017, and they found that derived forms of codfish living in variable concentrations of dissolved oxygen at the surface have more hemoglobin genes than ancestral codfish living in the constant environment of the deep ocean, which is very similar to what we see in the difference being between the cave comparing to the deep ocean and having that stable environment compared to the shallow ocean or surface watersheds having that more variable environment. So having that larger hemoglobin repertoire is advantageous in variable environments. And so when considering the less genes in the cave fish genotype, it's likely not due to lower concentrations of dissolved oxygen, but less variability in dissolved oxygen levels. Now, with the remaining genes, we decided to create a phylogenetic tree in which each orange circle is one cavefish gene and each blue circle is one surface gene. And this allowed us to organize the genes and allowed for more relevant comparisons, so we weren't wasting time comparing genes that weren't similar or didn't have any similarity. However, we did have to organize it a little bit more. Now the top blue box is what we called one-to-one nodes, where one cavefish gene lined up perfectly with one surface fish gene, and on the bottom we called these n-to-1 nodes, where one cavefish gene lined up to multiple surface fish genes. Now when we compared the genes within nodes, we found a few key things. Number one being 3 out of the 9 one-to-one nodes have nucleotide differences. All of the n-to-1 nodes have both nucleotide and amino acid differences. And each node with a nucleotide difference also had an amino acid alteration. Now, this is important because it means that no nodes had silent mutations. There are no silent mutations in these nodes. However, when we're talking about these alterations, we're not talking about just one or two. There are a high quantity of single amino acid alterations. And on top of these single amino acid alterations, we also see large deletions, specifically in this example. And these deletions and alterations are likely caused due to evolutionary pressure, which leads us to it all being connected. So you have this extreme environment with less dissolved oxygen and a more stable dissolved oxygen levels in general, which leads to this evolutionary pressure. And from this evolutionary pressure, we see things like the cavefish having less hemoglobin genes. And upon further review of the genes that still remain, we see a high level of genetic and amino acid alterations, which leads us to hypothesize that these alterations do cause a functional difference in the hemoglobin proteins that result, making them more likely to bind oxygen atoms efficiently. And at last, I'd like to thank the Gross Lab for having me.